and to reduce the spread of insect-borne diseases. This is the importance of insecticides. Hope you understand this slide. If there is any doubt at any point, you please feel free to clarify your doubts. Okay. And coming to the classification of insecticides, based upon the chemical composition, insecticides are classified into different groups. The first one is organochlorines, and the second one is organophosphates, and the third one is carbamates, fourth one is pyrethroids, and the fifth one is neonicotinoids, and the sixth one is biological insecticides. These are the things which we have classified based on the chemical composition. Okay, and coming to the first one, organochlorines. Examples of the organochlorines are DDT, that is dichloro-diphenyl trichloroethane, which is persistent in the environment but have been largely phased out due to environmental concerns. We are going to talk about these things in detail when we enter into botanicals and conventional insecticides. There is a separate unit for organochlorines, organophosphorus insecticides, carbamate insecticides, and botanical insecticides. Today, in this class, we are going to talk about the overview of the fundamentals of insecticide toxicology. So, don't feel bored and try to understand the things what we are learning today, so that we are going to study about all these things in detail in our future classes. Okay, so this is about the organochlorines, and the second one is organophosphates. Examples are malathion and parathion. These act by disrupting the nervous system of insects. That's how we to control the activity of the insects. And these chemicals are widely used in agriculture and public health to control the pests. And the third one is carbamates. Example is carbamyl, that is also called as 7, which targets the nervous system of the insects and which is used against a variety of pests to control and to protect the crop. Okay. And the fourth one is pyrethroids. There are synthetic versions of natural pyrethrins found in chrysanthemum flowers, that is chamanthi flowers, and which is known for their fast non-burn of insects. The example is permethrin. And the next one is neonicotinoids. The systemic insecticides like imidacloprid, which are observed by plants and protect them from sap-feeding insects. And the last one, sixth one, is biological insecticides, which are derived from natural materials. Whatever the things which we studied before are not the biological insecticides, but this is the biological insecticide. Example, from the Bacillus thuringiensis, we could do that, and from fungi, which target specific pests with minimal impact on non-target organisms. Nowadays, we are having, or we are in our day-to-day life, we are using Bt cotton. Yes, that is the resistance is from the gene isolated from Bacillus thuringiensis. Okay, so it is acting as a biological insecticide. That's how we would able to control. Now, what is the mode of action? There are two methods which you could see here. The first one is contact versus systemic action. Okay, here the contact insecticides means which will kill the insects upon direct contact. Okay, and which affects the nervous system or the cuticle. You know that the insects have the cuticle, which is a hard shell present on the surface. Okay, either it will affect the nervous system of the insect or the outer shell. And the second one is systemic insecticides, which are absorbed by the plants and translocated throughout their tissues and protects against pests which feed on them. And the other part is target sites in insects. You could see the target site. Already we discussed in our previous class that some of the chemicals are acting on the nervous system to kill the insects. Here also we mentioned the same. Target sites in insects. The first one is nervous system. We are targeting the nervous system of the insects to kill them in order to protect our crop. Okay. The first chemical is organophosphates, carbamates, and pyrethroids, which will disrupt the nerve function of the insect. And the second one is metabolic pathway. The insect's growth regulators and the biological insecticides interfere with insect hormones or physiological processes to inactivate and to control the pest replication or its development. In such a way, we could able to control the activity of the pests. Okay. And the next one is formulations of insecticides. Is that clear till here? Did you all understand all these slides which we have discussed till now?
Yes,
Hope
ma'am.
you understand, right? And the next part is formulations. This is the exact part which we have to study. Till now, we have studied about the introduction. Okay. And coming to the formulations of insecticides, there are three different types of formulations which we are going to study. The first one is liquid formulation, and the second one is solid formulation, and the third one is other formulations other than the liquid and solid. Okay. Coming to the first one, liquid formulation, emulsifiable concentrates. In short form, you can use EC also. These are oil-based formulations that mix easily with the water. Okay. And the suspension concentrates are the particle-based suspensions which require agitation. That means which require mixing. Agitation means mixing before use. And soluble concentrates are water-soluble formulations for easy mixing. These are the different types of the liquid formulations which you can use to control the pests. Okay, these are the chemicals which are given under the examples. And the solid formulations are dust, vegetable powders, and granules. Okay, coming to the dust, these are the fine powders applied to plant surfaces or into the cracks or crevices. And coming to the vegetable powders, these are dry formulations which will dissolve in water before application. And granules are pelleted formulations used for soil application. These are the solid formulations. We are talking about the formulations of insecticides. Okay, different types of formulations can be used to control the pests. The first one which we have studied are liquid formulations, and the second one is solid formulations. And now we are going to talk about gaseous formulations or other formulations. Under that, we are going to talk about baits, aerosols, microencapsulated formulations. Coming to the baits, which attract pests to ingest poison. And second one is aerosols, which pressurized liquids or gases for direct application. And the third one is microencapsulated formulations, which are active ingredients which are encapsulated for controlled release of the things. Okay, so all these are the formulations of the insecticides. Today we are talking about the introduction part. So make sure that you try to understand these things. We are going to talk about these things in depth in our future classes. And coming to the examples of the insecticides, we already studied these things in our previous slides. DDT, which is an example for organochlorine, malathion, which is an example for organophosphate, and tarbyl, which is an example for carbamate, permethrin, which is an example of pyrethroid, imidacloprid, which is an example of neonicotinoid, Bacillus thuringiensis, which is an example for biological insecticide. We covered these things in our previous classes. Here, out of these, some of the things like organophosphates, carbamates, and thyroid, these are the things which acts on the nervous system of the insects. Okay, this is what we studied. Okay, and coming to the safety and environmental considerations, some of the insecticides which we are using are causes risk to the humans and even to the wildlife. Coming to the environmental persistence, we concern about bioaccumulation and long-term effects on ecosystem. So whatever the things which we are using to control the pests should be used in a limited manner, okay? Not to harm the atmosphere, not to harm the human beings, not to harm even the insects also. Okay, we have to protect the ecosystem also. So make sure that we are using limited amount of the insecticides to control it. Maximum, we try to use the biological insecticides, not the chemical insecticides, to protect the crop or to protect any pests. Okay. And the next part is evaluation of insecticide toxicity. Okay. This is your second part of fundamentals of insecticide toxicology. Under this, we are going to talk about how do we evaluate the insecticide toxicity. Okay, how much we have to use, how to use, and all these things. Okay, and we already studied about the introduction, and we already studied about the definition of insecticide. Okay, as we are talking about the insecticide toxicity, once again we'll brush up the definition of insecticide. Insecticide toxicity refers to the ability of chemicals used to control pests to cause harm to organisms, including humans and non-target species. And its importance is understanding toxicity is crucial for ensuring safe agricultural practices, protecting biodiversity, and minimizing environmental impact. All these are very important. Without our knowledge, if you could use the insecticides in a large scale, not only it will damage the crop and the insects, it will also cause harm to the environment and the biodiversity. Okay, before we use this, we should know how to use, what to use, how much amount we have to use. Okay, and the three concepts which we are going to study in this evaluation of insecticide toxicity is we are going to talk about the lethal dose and lethal concentration, how much we have to use, and all these things. And here is the toxicity matrix. You know about LD50 and LC50. LD stands lethal dose. I already told you we should know how much we have to use. Okay, without our knowledge, we should not use in a higher concentration. Okay, LD stands lethal dose. LC stands lethal concentration. What is lethal dose? LD 50. The amount of substance required to cause death in 50% of a test population, that is called as lethal dose 50. We are talking about the toxicity metrics here. Here I have given you an example. Malathion is a chemical. It has an LD50. That means it has a toxicity matrix of LD50 of approximately 900 mg per kg in rats, indicating its acute toxicity. We should know about this. Okay, not only for this chemical, for all the chemicals we should know the toxicity metrics. And coming to the lethal concentration, the concentration of a substance in air or water that causes death in 50% of test organisms over a specified period. This is the difference between lethal dose and lethal concentration. The example is glyphosate or glyphosate, which has an LC50 of 361 mg per liter in fish, highlighting its potential impact on aquatic organisms. And the mode of action of insecticides, how it acts. Okay, it will interact with the target organisms to cause harm. Okay, and the examples are neurotoxic insecticides, and the second one is insect growth regulators. These are the insecticides which act on the nervous system of the insects. And example here given is organophosphates, which will inhibit acetylcholine esterase. Okay, we are going to talk about these things in our later classes. And the next one is insect growth regulators, which will disrupt the growth and developmental processes by acting on metabolic activities of the insects. Okay, so these are the examples. If you could see here, the mechanism, here you could see towards the left side, in the left panel, you could see 7 common nicotinoid pesticides, 1, 2, 3, 4, 5, 6, 7. Total 7 neonicotinoid pesticides are here. And towards the left side, you could see the mechanism to study neonicotinoid toxicity. Okay, how these neonicotinoids act on the receptors, okay, of the vertebrates and in the invertebrates. Okay, and how it causes damage to the insects. All these things will be given. Here, this is the receptor. If you could see my cursor, this is a receptor which you are going to see. Okay, the action of this neonicotinoids, when it acts on this, you could see the activity, the mechanism will be seen. The downstream signaling pathway will be affected such that it would able to control the pests or it would kill the insects. This is about uh, the mechanism. Here you could see ACH, which is called as acetylcholine, and here you are seeing NACHR, which is called as nicotinate acetylcholine receptor. In our previous slide, you have seen here um, organophosphates are the insecticide. I mean, we classified the insecticides chemically into different groups. Among that, one of the group is organophosphates, which will inhibit acetylcholine esterase enzyme. Okay, so this is about the mechanism. And the next one is environmental impact of insecticides. The first one is bioaccumulation, and the second one is persistence. What is bioaccumulation? How these insecticides shows its impact on environment. Say extra usage or high amount of insecticides may lead to bioaccumulation. What is bioaccumulation? Insecticides can accumulate in organisms over time, leading to higher concentration in higher trophic levels, okay, which is not good. And persistence, ability of insecticides to remain active in the environment, affecting soil, water, and wildlife long after application. Examples are DDT, which bioaccumulates in birds and disrupts reproductive cycles, leading to population declines. Okay, we are damaging the biodiversity or the ecosystem. Okay, this is about the impact of insecticides on environment. And impact of insecticides or the effect of insecticides on health and environment. Here I have given you two case studies. The first case study is human health impact and the second one is environmental effects. The first one is human health impact. In this, we are going to see pesticide poisoning in agricultural workers. In rural farming communities, frequent exposure to insecticides like organophosphates and carbamates during application can lead to acute poisoning incidents among agricultural workers because without wearing the mask, they will apply all these things to their crops at that time. Uh, you could see this impact. And the impact is symptoms range from mild irritation and nausea to severe respiratory distress and neurological effects. Long term exposure can contribute to chronic health conditions affecting livelihoods and community well being. Okay, sometimes you could see the damage in the DNA also. Due to the damage in the DNA, it may cause many different types of deathful diseases also. Not only it affects the neurons or it affects the nervous system and the lungs or the respiratory system, it will also affect the DNA. So in maximum number of the cases, you could see this, the DNA gets damaged. You could see the fragmentation of the DNA that ultimately leads to sometimes the death of a person also. Okay, and the second aspect is environmental effects. Here, decline in bee populations due to neonicotinoids. Okay, you know about bees and you know what are neonicotinoids also. Okay, neonicotinoid insecticides used widely in agriculture to protect crops from pests have been implicated in decline of bee populations worldwide. These systemic insecticides are observed by plants and can persist in pollen and nectar, leading to sublethal effects on bees such as impaired navigation, reduced foraging efficiency, and compromised immune systems. This ultimately threatens pollination services critical for crop production and ecosystem health. Okay, these are the effects of using insecticides, both on environment and on human health. Come to the next part, insecticides and cuticle interaction. You know what, till now, what all we have studied are formulations and classification of insecticides. There are three formulations: liquid, solid, and gaseous. And we have studied about the classifications where we have studied carbamates, pyrethroids, and organophosphates, organochlorines, biological insecticides. All these we have studied based on the chemical. Uh, uh, we divided the insecticides into 6 different groups, and even we completed the evaluation of insecticide toxicity. Okay, how much we have to use. If we are using more, what is the impact of the insecticides on human and uh, environment? These are the things which we have studied. And now we are going to talk about insecticides and cuticle interaction. Did you all understand what we have learned till now?
Yes, madam.
Okay. The next one is insecticides and cuticle. The first one, if you could see the definition, it will describe the insect cuticle, which acts as a protective outer layer, which is very thick, composed of chitin, proteins, and lipids. This is the composition of the cuticle layer, which you are seeing on the insects, which is the outermost layer. And it helps in providing structural support to the insect, and it will protect against physical damage, and it will regulate the water balance too. And coming to the structure of the insect cuticle, you could see, if you could see the structure, it has 3 layers: epicuticle, exocuticle, and endocuticle. Epicuticle is the outermost layer. If you could see the section of this cuticle, epicuticle is the outermost layer, exo means it is below that, and endo means it is the inside layer. It has 3 layers, and there uh, it uh, it has 3 layers, and it is made up of, as I already told you, um, proteins and lipids and chitin also. And there is an example which you are seeing here. The example is uh, honeybees or bees. Honeybees have a complex cuticle structure which protects the bees from environmental stress and chemical exposures like neonicotinoids. Okay, because this is an example of insecticide. What is the mechanism of insecticide penetration? You could see the mechanism here: cuticular permeability. How this cuticle structure will be, how the composition influence the penetration of insecticides into the insects. All these things to be studied, that we are going to study in our later classes. And the factors which affects the permeability. Here, we are going to study about the role of the lipids, and I already told you cuticle is made up of chitin, lipids, and proteins. So we are going to talk about the importance of lipids, wax layers, and cuticular hydrocarbons in hindering or facilitating insecticide penetration. And coming to the pathways, we are going to study about the routes through which insecticides can enter the insect body, either by contact or by ingestion. Examples are fruit flies or house flies. Okay, fruit flies typically have a thinner cuticle with higher permeability compared to house flies, which affects their susceptibility to insecticides like pyrethroids. Coming to the types of insecticides and the mode of action, we already studied this in our previous slides. That is contact and systemate insecticides. Okay, and we already studied the mode of action also. Some of the insecticides act on the nervous system of the insects. Some of the insecticides act on the metabolic processes of the insects, and some of the insecticides directly act on the growth of the insects. Okay, so the examples are neonicotinoids. And coming to the next part, we have to study about the cuticular resistance and insecticide tolerance. We are going to study about the mechanisms of resistance, examples. Under the mechanisms of resistance, we are going to study how the insects can develop resistance to insecticides through changes in cuticular structure, metabolism, or target site insensitivity. Coming to the examples of resistance, we are going to discuss about the case studies where insect populations have developed resistance to specific insecticides due to mutations or physiological adaptations. Nowadays, due to continuous spraying of the insecticides to the crops, the insects also develop resistance towards that particular chemical. At that time, you cannot kill that insect. Okay, it happens either through the continuous usage of the same chemical years long, or through the climatic conditions. This will happen through these two things. Okay, and the next one is the implication. We are going to highlight the challenges in managing resistant pest populations and the importance of integrated pest management strategies. Examples are bedbugs. You have an idea about the bedbugs. Certain bedbug populations have developed cuticular resistance to pyrethroid insecticides, contributing to the resurgence of infestations globally. And the next one is environmental and ecological impacts. If you could see this, there are several things which we have to concentrate, non-target effects. We are going to discuss about unintended consequences of insecticide use on beneficial insects, pollinators, soil organisms, and aquatic ecosystems. And the next one is persistence and bioaccumulation. We are going to talk how insecticides can persist in the environment, accumulate in food chains, and impair wildlife and human health. Examples are pollinators. The use of neonicotinoid insecticides has been associated with declines in bee populations due to sublethal effects on foraging behavior and colony health. Okay. And what are the strategies that must be followed for sustainable pest management? We have to go for integrated pest management. We have to use alternative control measures. Okay. And we have to know about community engagement, where we have to study the importance of educating farmers and stakeholders on sustainable pest management practices. Example, IPM programs, which will showcase the successful IPM programs that have reduced reliance on insecticides while maintaining crop productivity and environmental health. Okay. By this, we completed the overview of the fundamentals of insecticide toxicology. Yes, did you all understand?
Yes,
Yes,
madam.
ma'am. Yes, now we are going to talk about, can you give me a time, please? We'll talk about the first one, botanical insecticides. Okay.
Yes, madam.
Hope you understand the introduction part which we discussed today.
Yes, madam.
Can you see the screen? If you could see the screen, please let me know.
No, ma'am.
No, madam.
Okay, 1 minute. Yes, can you see the screen now?
Yes, madam.
Okay. Okay. Hope you understand what all we discussed till now. Okay. And now we'll start with botanical insecticides. Okay. When we classify the insecticides among all, one of the insecticide is botanical insecticide. We have studied in our previous slides. And now we'll study about the botanical insecticides in a deeper way. What are botanical insecticides? These are naturally occurring chemicals extracted from seeds, flowers, leaves, stem, and roots. Okay, it is called as botanical insecticide, or they are called as botanical insecticides. The name itself is indicating it is from living organisms. Okay, especially from the plant products, plant materials. Okay, you could see the pictures also here. Okay, and 1 minute, please. Okay. Can you see the screen now?
Yes,
Yes,
madam.
Yes. So you understood that what are botanical insecticides? These are naturally occurring chemicals and extracted from the seeds, flowers, leaves, stem, and roots of the plant. Okay. What are botanical insecticides? Insect toxins derived from certain plants. You could see the quick action on target pests. Okay. And you even see, okay, I'll send you the material. Okay. Quick action on target pests, broad spectrum action, do not persist in the environment. I'll tell you the important thing here. Why we are focusing on botanical insecticides? Because they are derived from the plants. They are derived from the naturally available living organisms. Okay. It does not cause any harm to the human beings, to the environment, even to the ecosystem also. Okay, which will help us to control the pests, to protect our crop, not only to protect our crop, uh, to protect the human health also. Sometimes some of the pests like mosquitoes, all this cause damage to the human health also, which causes malaria and dengue also. Okay, these are also comes under the pests or the insects. Okay, we could able to control all these by using the botanical insecticides. Okay, we are going to talk about in depth about the botanical insecticides. What are the factors which affects the use of botanical insecticides? The first one is the raw material availability, and the second one is the solvent types, plant species, and part of plant. Third one is rapid degradation, and the fourth one is state registration. Fifth one is market opportunities for botanical pesticides, and the sixth one is weather conditions. These are the factors which affects the use of botanical pesticides. Okay, we have to be careful when we are using the botanical pesticides or insecticides. If you could see the advantages of these things, often these plants also have other uses like household insect or plants with medicinal applications. Some of these products may be used shortly before harvesting. Many of these products act very quickly inhibiting insect feeding. Since most of these products have a stomach action and are rapidly decomposed, they may be more selective to insect pests. Most of these compounds are not phytotoxic. These are the advantages of botanical pesticides. When we are going to talk about the organochlorines, organophosphorus insecticides, and carbamate insecticides, and syntheroids, and other chemical insecticides, differences and the similarities of all insecticides not to use. To which insecticide we have to give the priority. Okay. But as we are dealing with only botanical insecticides, just we are highlighting the advantages of botanical insecticides. These are not phytotoxic. Okay. These plays a major important role in protecting the crop, and these can be used shortly before harvesting. Okay. These are quickly inhibiting insect feeding. Okay, these are the importances or the advantages of the botanical insecticide. If you could see the disadvantages, many of these products are not truly pesticides because they are merely insect deterrents and their effect is very slow. They are rapidly degraded by UV light so that their residual action is very short. They are not necessarily available season long. There are no legal registrations establishing their use. Not all recommendations followed by growers have been scientifically verified because they don't have that awareness. And in the next, we are going to talk about some botanical insecticides. We could divide the botanical insecticides into two generations. In the first generation, you could see nicotine, protein, and rhinin, pyrethrin. And the second generation includes synthetic pyrethroids and neem products. And if you could see the potential new botanicals, ananios, acetogenins, and the second one is suproestes. Okay. If you could see the botanical pesticides, we could divide into two generations. Okay. Depends upon their usage. We divided this in depends upon the usage. And the first one is nicotine. What is nicotine? This is an alkaloid which we could get from a tobacco plant, and its scientific name is Nicotiana tobatum. Nicotiana tobatum. Okay, and this nicotine, which is an alkaloid derived or extracted from the leaves of tobacco plant, and it shows its action and acts on fat strips and this is the function of this first generation botanical insecticide, nicotine. Okay, and if you could see the market product of it is called as nicotine sulfate, which is, we already know that it is a botanical insecticide derived from the tobacco plant. That is from, it is a plant, it is from plant origin. It is very effective against a wide range of insect pests affecting crops of economic importance as well as on ectoparasites affecting livestock. And the mode of action, if you could see the mode of action, this nicotine sulfate, as I already told you, it is available in the form of nicotine sulfate. It acts upon the central nervous system of the insects. It has both direct contact as well as the fumigation action. And if you could see the use of this nicotine sulfate, 40% of the nicotine sulfate is used to kill aphids, bugs, worms, leaf hoppers, and similar certain insects which attack and destroy fruits, vegetables, crops, and even flowers. It is also effective against lice, mites, and ticks, which are a menace to livestock. This is the use of nicotine sulfate in protecting the leaves or the crops. This is the thing which you could get it from the market. 40% of nicotine sulfate. It is a botanical pesticide, dead, isolated or extracted from tobacco plant. Okay. And the second biological insecticide is pyrethrin. What is pyrethrin? It is also a botanical, one of the botanical insecticide from the first generation derived from chrysanthemum, and we should not mix with the soap here. And the examples of pyrethrin is pydanate, and it contains 5% of emulsifiable concentrate. We already studied these things. What are emulsifiable concentrates? All these things in our previous slides under the introduction part. Next, pydanate contains 5% EC and omri and evergreen. Okay, and which is extracted from the flower of chrysanthemum. You could see the flowers which is given in the picture. Okay, and its action is stomach poison. Okay, and it affects on caterpillars, aphids, leaf hoppers, spider mites, bugs, cabbage worms, beetles. Okay, and if you could see the picture here, maybe you could, maybe it's not here, but you can see here pyrethrin 5EC. Okay. If you could see the market product, it is naturally derived product from the flowers of chrysanthemum, and it is professional grade pesticide which attacks the nervous system of a wide spectrum of insects. And pyrethrin 5EC kills aphids, fruit flies, fungus, and many other insects. Pyrethrin 5EC is registered as suitable for organic and non-organic farming as it kills pests quickly, leaves no toxic residue, and edible food crops can be safely harvested only 24 hours after application. Pyrethrin 5EC is a natural insecticide made from dried chrysanthemum flowers, and it is registered as suitable for organic purpose. And the target application is, if you could see, we already know this pyrethrin targets different types of the insects listed here, and it is used against small sized and immature insects. Okay, and you could add some of the synergies like PBO to increase the efficiency, okay, efficacy of it. And the third one is the neem. Okay, we already know it is Treaty of India. I mean, like it is from Azadirachta. The scientific name is Azadirachta indica. Okay, In India, 14 million trees, of which 50% of the Azadi rectangulate or the neem trees are seen in UP. Though not a forest tree, it grows wild in the forests of AP, Tamil Nadu, and Karnataka. Fully grown neem tree yields 50 kg fruit annually and 350 kg of leaves. India probably produces 0.7 million tons of fruits and 5 million tons of leaves every year. This is the introduction of the neem. Almost every part of the tree is bitter, as we all know, and finds its application in indigenous medicine. Oil and cosmetic industries are the users of neem. The refined and purified neem seed oil has many therapeutic properties. Considerable quantities of oil are used in cosmetic preparations. Nowadays, people are using neem oil uh, to control the pests. Neem oil must be sprayed to the crops to control the insects. Okay. Considerable quantities of oil are used in cosmetic preparations. The neem cake after oil recovery is used for slow release of nitrogenous fertilizers. You can use the neem leaves powder as a fertilizer. Neem oil contains limonoids, a class of compounds that act as antioxidants or growth regulators in insects. They do not kill instantly, but wipe out a whole generation of insects by preventing the young ones from maturing and the adults from reproducing. We could control the pests. And the neem oil contains limonoids, as we already studied in our previous slide, a class of compounds which acts as antioxidants or growth regulators in insects. It is similar to the insect hormone edusone, which is needed for molting during insect development. It works at a concentration of 1 to 10 ppm by blocking edosine action, thereby preventing the larvae from shedding their external skeletons and maturing. It also prevents feeding in about 200 insects at a concentration of 10 to 100 ppm. This is the product Azadiratta from Azadiratta Indita. It is also called as Azadirattin, isolated, extracted from the neem trees. Parts of the neem trees, especially from the seed or the fruit. It acts as a systemic poison. It shows its effect on stem borers, bollworms, leaf miners, caterpillars, etc. Triple action neem oil is available in the market, which is an organic way to protect the plants and which is extracted to protect the plants from all different types of the diseases like caterpillars, mildews, leafhoppers, etc. This can be a big problem throughout the nation, so make sure to protect all your plants by using this particular triple action neem oil. Okay, it is not a contact poison. It does not kill the insect pests directly. It is a systemic inaction, mainly affecting the feeding as well as growth and development of insects. Nothing will happen immediately, even if you spray the oil on them. So people often come to the conclusion that neem oil is ineffective and just all type. But in our previous slide, we have studied that it is slow in action. Okay, it does not kill instantly, but wipe out a whole generation of insects by preventing the young ones from maturing and the adults from reproducing. This is the action of the neem oil, which we have isolated or extracted from the seed or the fruit of the neem. And the next one is nicotinoids. Okay. And coming to the nicotinoids, we already studied what is nicotine in our previous slides, which was isolated from, which is an alkaloid isolated from the tobacco plant. Okay. We'll see about this. Nicotine represents a class of compounds, nicotinoids, with biological activity, which is mainly due to interaction with acetylcholine receptors. Use of nicotine sprays is banned in India, restricted use in USA to home gardens and some greenhouse cultures. It is due to nicotine has a fairly high mammalian toxicity. It has a narrow insecticidal spectrum. It has a very short lifespan and low residual activity under field conditions. And it is metabolized and excreted fairly fast in insects. If you could see the advantages of the nicotinoids, it acts on a different target in our system of the insects. Hence, insect strains which develop resistance to Acetylcholine esterase insecticides are not likely to develop cross resistance to nicotine, which will develop a new generation of synthetic insecticides, which are called as neonicotinoids. Coming to the properties of physical and chemical properties of nicotine. In 1690, water extracts of tobacco leaves were used to control certain insects on garden plants in Europe. In 1734, nicotine is used to control of plum turquillo due to the presence of the alkaloid nicotine present in tobacco leaves and named after Jean Nicot in 1829. Okay, and nicotine L1-methyl 2-pyroldine or L3-pyridine is the principal alkaloid in the leaves of Nicotiana tobatum, and L rustita to the extent of 2 to 8%. These are the chemical names of this particular alkaloid. Two other closely related compounds, that is nornitrotin and anabasine, are present in these plants, also have some insecticidal activity. They act as nerve poisons in both insects and mammals. Non-nicotine predominates in Nicotiana sylvestris and anabasine in Anabasis aphylla. There are several other naturally occurring nicotinoids like tonin, which is poison of hemlock, cystine, which is an alkaloid of laburnum, anatoxin A, which anatoxin of freshwater blue green algae, Anabina flostre, labolin, which is active ingredient of Indian tobacco. These are the properties which we are listing now. Free nicotine is colorless or pale yellow oily liquid, acrid burning taste and odor of pyridine, and miscible with water below 60 degrees Celsius, highly soluble in alcohol, chloroform, ether, kerosene, and oils, highly hygroscopic and turns brown and viscous on exposure to air or light and develops a disagreeable odor. Nicotine sulfate is stable and freely soluble in water and alcohol. Free nicotine is highly volatile and used as fumigant in greenhouses. Nicotine sulfate acts as a stomach poison for leaf-eating insects. It releases free nicotine on spraying. Addition of alkaline compounds such as soap and calcium cysteine liberates free nicotine, which acts as a contact insecticide or as a fumigant against soft body insects. Nicotine is highly toxic. Its oral LD50 for rat is 30 mg per kg. It is rapidly detoxified and eliminated from animal body. In the presence of light, it decomposes easily. Nicotine is used as a sulfate representing 40% of actual nicotine. Nicotine sulfate is not volatile and is less to man than free nicotine. All these are the properties of the nicotine, both physical and chemical properties. Coming to the structure toxicity relationship, it has the pyridine ring. I'll show you the picture when I come to the next slide. Maybe it is in another PPT. Okay. We'll go through these things. I'll show you the structure later. Coming to the structure of nicotine, you could see on the pyridine ring, and the distance between the two nitrogen atoms should approximately be the distance between the two functional groups of acetylcholine. That is an optimum distance of about 4.2 angstroms between the two nitrogen atoms and unsubstituted alpha position of the pyridine ring. If I show you the picture, then that would be easy for you to understand all these things. I'll show you the picture. I'll try to open the other PPT after I complete this part. Coming to the mode of action, we already studied about in introduction, we used the word acetylcholine receptor. Okay, acetylcholine receptors are classified into two classes based on sensitivity. The first one is agonist and the second one is antagonist. The drugs whose binding to acetylcholine receptors mimics the effects of acetylcholine, and the antagonists are the drugs whose binding to the receptor inhibits its response to acetylcholine. And coming to the other part, the nicotinate acetylcholine receptor found in autonomic ganglia, skeletal muscles, and brain, which serves as molecular target for nicotine, activated by nicotine and inhibited by purine. The muscarinic receptors found in smooth muscles, cardiac muscles, autonomic ganglia, glands, and brain, activated by L-muscarine and inhibited by atropine. Snake venom toxin, alpha bendarotoxin, is a highly specific antagonist of the nicotinate acetylcholine receptors of skeletal muscle end plates. Purine is a competitive antagonist at muscle end plates and is only a channel blocker or non-competitive inhibitor in autonomic ganglia. Lobeline has a nicotine light stimulant effect followed by inhibition in autonomic ganglia, inhibits transmission without stimulation. Acetylcholine is a neurotransmitter in central nervous system of insects. Nicotine is an agonist and mimics a part of the action of acetylcholine by interacting with nicotinate acetylcholine receptor. The effects produced by nicotine across the synapses and neuromuscular functions in mammals are called nicotinate effects. That is the reason it is banned in India and there are some restrictions in other countries too. In mammals, the primary effects of nicotine are at neuromuscular junctions and at the cholinergic junctions such as smooth muscles and glands. In insects, the effects are on ganglia of CNS, central nervous system, because the insect neuromuscular junction is not cholinergic. Both acetylcholine and nicotine produce stimulation at low concentrations and blockage at higher.
Excuse me, ma'am.
Stimulation.
Voice is not audible. It is low.
Can you hear me?
Yes, ma'am. It's now okay.
Okay, I'm sorry. From where should I continue?
From the second point, ma'am, in insects.
In insects, the effects are on ganglia of central nervous system because the insect neuromuscular junction is not cholinergic. Both acetylcholine and nicotine produce stimulation at low concentrations and blockage at high concentrations. First stimulation and then blockage, so that early twitching and convulsions followed by paralysis. You will see all these things in the insects. Nicotine has no inhibitory effect on cholinesterase activity. However, at higher concentrations, the enzyme is inhibited by nicotine. According to Yamamoto, the free base penetrates easily through the integument, while nicotinium ion is slow to penetrate due to the presence of positive charge on the pyrrolidine end. After reaching the body fluid, an equilibrium is established between the two at the pH of the hemolymph in insect body. After penetration through nerve barrier, a new equilibrium is established between the ion and the free base. The nicotinium ion is the physiologically active entity and it interacts with acetylcholine and hence you could see the insecticidal activity. Coming to the effect of this nicotine on the autonomic nervous system. Nicotine affects the functioning of the autonomic nervous system of mammals and the effector organs, which receives both sympathetic and parasympathetic innervation. It has both stimulant and depressant phases of action. Nicotine may increase the rate of heartbeat by excitation of sympathetic or depression of parasympathetic cardiac ganglia. Nicotine causes increased discharge of acetylcholamines from the adrenal medulla. Catecholamines accelerate rate of heartbeat and raise blood pressure. The effects on gastrointestinal tract are due to parasympathetic stimulation. transmitter of all preganglionic nerve cells whose cell bodies reside in brain or spinal cord and whose axon terminate and for synapses with ganglion cells in the autonomic ganglia. Nicotine depolarizes ganglion cell membranes, thus acting as an agonist to acetylcholine at low doses. If it persists due to slower degradation or excretion, or when high doses are administered, it causes depression and total blockage of nicotinate cholinergic synapses of autonomic ganglia. These effects are a result of acetylcholine receptor activation at low doses and ACH acetylcholine receptor desensitization at higher doses. This is all about the effects of the nicotine. And coming to the effects of the nicotine on central nervous system. Till now, we have studied the mode of action and the effects of nicotine on autonomic nervous system. And now we are going to talk about the effects on the central nervous system. Like how you have seen the effects of nicotine on autonomic nervous system. Likewise, you could see the effect of nicotine on central nervous system too. In small doses, it acts as a stimulant, but in large doses, it acts as depressant. The respiratory centers of the brain cells are stimulated at small doses and depressed at higher doses. Respiratory failure occurs with nicotine poisoning, but paralysis of central respiratory centers as a late result of nicotine poisoning occurs after the peripheral career-like effect on the respiratory muscles. Tremor is one of the more characteristic effects of nicotine in humans and in other mammals. Although nicotine has demonstrated effect directly on skeletal muscle, nicotine tremor is undoubtedly a result of central nervous system stimulation. Fatal doses of nicotine generally cause convulsions in all mammals, which due to stimulation of the male brain rather than the spinal cord, since they are clonic rather than tonic in character. Convulsions induced by nicotine differ from convulsions produced by strychnine or pentylenterozole and are blocked by selective group of drugs for their anticholinergic activities, but not by cholinergic antagonists. Nausea and vomiting are the most common symptoms of acute nicotine poisoning, and administration of even very small amounts of nicotine causes nausea and vomiting. Small doses of nicotine are also emitted. The major site of action is the chemoreceptor trigger zone in the brain. These are the effects of central nervous system. Coming to the effects of nicotine on neuromuscular transmission. Nicotine effect on skeletal muscles, which is it is the one that is actually life-threatening in mammals. If injected in the fraud, nicotine produced fibrillary twitching and slow prolonged contraction of the skeletal muscles, which were not prevented by previous division of the motor nerves, innervating those muscles but disappeared upon injection of the antagonist urate. On the other hand, the paralysis induced by urate was partially reversed by small quantities of nicotine. Similar effects were observed in reptiles, birds, and mammals. Thus, large doses of nicotine cause muscle paralysis like urate. Nicotine depolarizes the neuromuscular postsynaptic membrane by reacting with the acetylcholine receptor like an agonist and causes muscle twitching. The synaptic blot resulting in muscle paralysis occurs in two stages. The initial stage is due to depolarization, and the second stage is the latent but more persistent blot after repolarization. Interactions of acetylcholine or nicotine with acetylcholine receptor reflect the presence of two affinities, a lower affinity representing the activated receptor state and a higher affinity representing the desensitized state. At higher concentrations, nicotine binds to sites on the ionic channel moiety of the receptor protein and depresses ion transport. The channel blotting activity is a property of drugs that acts as non-competitive inhibitors of neuromuscular transmission. Coming to the last one, toxicity to mammals. For higher mammals, nicotine is one of the most rapid and toxic poisons known. The fatal dose of nicotine for an adult is less than 1 mg per kg, and death can occur in 5 to 30 minutes. Absorbed rapidly from time to time, from stomach because of acidity of grape juices. Free nicotine is absorbed through unbroken skin. The onset of symptoms include nausea, vomiting, salivation, abdominal pain, etc. And all these things are due to the effects on central nervous system and parasympathetic. Nicotine and its metabolites accumulate in and depart from the brain very rapidly. Nicotine or its metabolites also concentrate in the adrenal medulla, superior cervical ganglia, gastric mucosa, and walls of blood vessels. It is metabolized effectively to less toxic metabolites and is eliminated fairly fast. The cause of death is usually the result of respiratory failure, that is caused by paralysis of the respiratory muscles. If respiration is restored, convulsions stop, then the animal survives. So we have studied about both these things, effects on the autonomic nervous system, and we studied on the central nervous system, neuromuscular nervous system, and toxicity to the mammals. And the next part is toxicity to the insects. Nicotine is considered as a contact insecticide and also acts as a fumigant. One mode of entry of nicotine into the insect is through the spiracles into the tracheal system. Symptoms include excitation, convulsions, paralysis, and death by affecting CNS. Nicotine has a profound effect on the function of heart in insects. Coming to the molecular targets, this nicotinate cholinergic receptors of the CNS of insects are primarily targets of nicotine action and also through blockage of acetylcholine receptor channel or receptor or channel. Other targets in invertebrates may be axons, and nicotine have a local anesthetic light action on squid giant action. Coming to the metabolism, oxidation occurs on 5' carbon atoms and nitrogen atom and on N-methyl group of nicotine to form nicotine, protein 1 oxide or non-nicotine respectively. I'm going to show you all the structures when I show you the next PPT. I'm going to show you the structure of nicotine, and if you could understand the structure of nicotine, it will be easy for you to understand how the metabolism occurs and all. Methylation on the pyridyl nitrogen atoms also occur to give nicotine isomethionine. Soil bacteria oxidize nicotine on 6 carbon to form 6 hydroxynicotine. Major metabolic product was found to be nicotinin and eventually 3-pyridyl acetic acid. There are two ways by which insects escape from toxic effects of nicotine. Some metabolize nicotine to avoid intoxication, while others do not metabolize nicotine but excrete into feces. So this is all about the effect of the nicotine on different areas and how the organisms escape from the action of the nicotine. And the final one is the therapy. To treat the nicotine poisoning, and the second one is artificial respiration until body eliminates sufficient quantities of the drug. And before there is a severe drop in blood pressure, if drug is swallowed, gastric lavage is helpful. Introduction of slurry of activated charcoal into the stomach is also acts as a therapy used in therapy. Vomiting should be induced, treated with anticonvulsions because convulsions are the symptoms which you are going to see. Paralysis, convulsions, all these things you could see with the effect of the nicotine. By this, we completed the first part, nicotine, which we extracted from tobacco plant. This is the second one, which we are going to study this in our next class, that is tomorrow's class. Before we enter into the next topic, we have to complete the structure of nicotine and how the metabolism occurs and all. Okay, by this we completed today's class. Hope you understand what we have studied along with the introduction. It is when we have studied the introduction, it is an overview, and we have studied this nicotine part today. But in our next class, we are going to talk about the structure of nicotine and other aspects before we enter into the new topic. Hope you understand this. Is it so?
Madam, can you please share this PPT in the group, ma'am?
Group means in a Zoom?
WhatsApp group.
I'll share. Okay.
Okay, ma'am.
In the WhatsApp group, I'll share the PPT.
Okay, ma'am. Thank you.
Can I share you tomorrow so that I have to add few of the structures here? Once I'll add the structures, I'll share the PPT in a group.
Okay, ma'am. No problem.
Okay, ma'am.
Okay,
Thank you.
because we missed a few of the things here.
Okay, ma'am.
Okay, it is as it is your first class, I thought I could complete only the introduction. Okay, that
Okay.
is the reason I didn't add those things. Okay,
Okay,
once
ma'am.
with that, we'll share the PPT in a group. Clear?
Yes, ma'am. Thank you.
Okay, ma'am. Thank you. Thank you all for your patience hearing. Shall we leave for the day?
Yes, ma'am.
Okay, ma'am. Thank you.
Thank you.